So now our second speaker in this part is going to be Professor Kathryn, and she's been awarded a Project for Translation Award in 2021. So for those of you who don't know about this award, this is the predecessor to the Confidence for Projects Award that was presented by Nigel this morning. And so Kathryn is going to speak to us about her work on discovering and assessing new pharmaceutical drugs aimed at eliminating morbidity from the whipworm.
Okay, so thank you very much for the invitation to speak this morning. What I thought I would do is use the first couple of slides to introduce you to the disease, which is the target of our novel drug discovery research, and then spend the rest of the time picking out a few of the highlights from our Project for Translation Award. So, neglected tropical diseases, really. Whatever way we try and measure health, neglected tropical diseases are a major cause of health inequality. So they affect low and low to middle income countries disproportionately, and they disadvantage people and whole countries. They are diseases of poverty, so they affect the poorest of the poor on the planet. And current drugs to treat anti-neglected tropical diseases are becoming ineffective against these parasites due to the development of parasite resistance. And it's worth noting that the current anti-neglected tropical disease drugs were primarily developed in the 1970s for veterinary indications, and they've been sort of co-opted over time for use in humans. So novel drugs are urgently needed. Soil-transmitted helminths sit under the umbrella of neglected tropical diseases, and the soil-transmitted helminth we're particularly interested in is the whipworm, which is shown on the right-hand side of this slide. And in countries such as Madagascar and the Cameroon, which is where we've done some work, this whipworm can reach up to 90% prevalence. So shockingly prevalent. So introducing the whipworm. So the human species of whipworm is Trichuris trichiura, infects around 500 million people globally. There are three major soil-transmitted nematode parasites. So these include hookworm and ascaris, as well as our whipworm. Tends to cause morbidity, not mortality. So it will present as growth stunting, anemia, child growth delay. And in the laboratory, we're using a preclinical model of human trichuriasis, Trichuris muris in the gut. And this is cecal-dwelling nematode of mice. It's a great model of human trichuriasis. And it is partly intracellular, living in the large intestine. So this is fascinating in terms of the worm biology. It's a really unusual niche for something that is sort of 2 to 3 centimetres big. And it's worth pausing on those 2 points for a minute, because they do present drug development challenges. So this is the front end of the worm, and if it burrows in the gut epithelial cells, so it forms a tunnel on gut mucosa, which is shown here. And if I were to take a transverse section across here, this is what we'd see. So this is the head of the worm sitting within the enterocytes, which line the gut. And again shown here in this histology image, that's the front end just snaking its way through the epithelial cells. Current drug treatment strategies focus on killing the adult worm within the host, and that's also the target for our drug discovery, although we do have this aspirational idea that we'd like to develop an environmental treatment which targets the eggs in the soil, so it would actually prevent transmission in the first place. The main drugs that are used currently are albendazole and mebendazole, so these are benzimidazoles. And if I can just draw your attention to the cure rate, which is shown in the column second to the last on the right-hand side, these are shockingly low. So 30% cure rate. It's simply not good enough in order to allow the World Health Organization to reach their 2030 target of reducing morbidity. Around 700 million children receive these drugs annually, either as a single dose once or twice a year. So we are a multidisciplinary team. There's myself and Dr. Ruth Foreman, who's in the audience, based at Manchester, and we've been working with medicinal chemists at Oxford, Angela Russell and her postdoc Carol Bataille, and we've been working with UCL with Professor David Sattel and Freddie Partridge. We've been working together since 2014, and we screened a number of compound libraries, and one in particular looked really, really good, and we called it the IMP series. It had a marvellous EC50, so very effective against the worm in vitro, and importantly it also worked against benzimidazole-resistant nematodes. So with that proof of concept lead compound, we wrote a pre-proposal to the MRC DPFS scheme in 2020, and that was declined. And it was declined because the panel felt that our lead compound had got unacceptable HERG activity. So what is HERG? Well, HERG encodes a potassium channel in the heart, and when that channel gets blocked, basically your heart could move into arrhythmia and you could suddenly die. And a number of drugs have been withdrawn from late clinical trial stages because of HERG activity. So this is our block. We needed to identify a lead compound which was effective against the whipworm and had acceptable HERG. So we were sitting at D3 in the translational pathway. We had this initial proof of concept lead compound, and we needed to establish a revised compound which had a better safety profile. But we had no money, no hands and no time, so we applied for a project for Translation Award, which we were successful. And this allowed us to move through and screen more members of the IMP pathway and also of the related compound series, the IMPAs. This is how our screening works, which I think is worth just showing you. So we call it an Invap setup, but basically there's a camera, there's a light source at the top, and then the worms sit in a plate. And it's a really powerful system because it's automated, it's high throughput, it allows us to screen drugs very, 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 very quickly and in a sort of agnostic way. So we measure log pixel variance over time. The blue line shows you the average pixel variance. The green line is average pixel variance plus 1 SD. So the blue bars are highly motile worms. And this is what that would look like on an image 96 well plate. So that's our screening system. And using that screening system, we then went on to try and identify advanced lead compounds which had a better safety spec. And I must say there's a huge amount of work that sits behind this one slide. But the end result on the right-hand side, we identified the IMPAs, which is a related compound series to our original compound series, excellent EC50s, much better acceptable HERG profile, good cytotoxicity, and we did lots of other ADME tests and safety profiling just to make sure that those drugs looked fit for purpose. So we've moved from the left with this OXO 7417, which had this red flag, the HERG, to the right-hand side where we've got a number of compounds now that have a good worm EC50 and a better safety profile. So armed with that information, we then reapplied to the MRC DPFS scheme. We put a pre-proposal in 2021, and this time we were successful, and so we were invited to full proposal in 2022. The other thing that the Project for Translation Award did for us was to allow us to establish this new collaboration with Katie Moore in Material Sciences and Nick Lockyer in Chemistry. And we also benefited from a research development fund for collaboration, which was awarded to Ruth Foreman to build on this technique. So we were using their methodologies, nanosims and TOFSIMs, which allows us to image drugs both at the cellular and subcellular level to determine the localization of drugs in tissues. And really, I can't think of any other way we can do this. This is a real problem if you're trying to develop drugs that work in the gut in vivo. And capturing those sort of drug biology behavior is quite tricky. So we're very excited by that collaboration, and it's very much ongoing. So I've mentioned Oxford and UCL as I've been going along. I would also like to say a massive help to the Translation Manchester team, and particularly Alessandro and Fiona, who were terrific in our preparation for that full proposal to the MRC DPFS. Deb with the impact, and also Duncan Henderson helped us with the prior arts searches. And Sam, I don't know if Sam's here, but Sam Butterworth peer-reviewed our proposals, both at pre-proposal and full proposal stage. So that was a massive help, and I would strongly encourage anyone to use the sort of network of successful people to get feedback on proposals that are going in the future. So that's it. Thank you.